which is a research unit part of INRAE, and we are also part of Université Paris-Saclay, which is in the Paris area, so in the southwest of Paris, near Versailles. We are around 300 people working on microbiology, and we work especially in pathogenetic microorganisms, the study of the microbiota, and we also have a pool, which I'm part of, which is about synthetic and system biology. So mainly biologists. So my work since my PhD in 2013 is around the understanding of the relationship between the protein production of the bacteria physiology. And now I'm working on cell-free system where there is no more bacterial physiology, but it's a useful tool to understand it. And so I did a postdoc in US, one in UK, then in Pasteur, and then I came back at INRAE where I have a permanent position. So what about the presentation today? So the maximization of production in cell-free. So first, I will do an introduction of what is cell-free system. Then I will talk to you about past work that I did about the exploration of the cell-free composition to maximize protein production using active learning. And then a work I'm doing right now with my PhD student, Léa Wagner, on the exploration of the cell-free composition, but this time to maximize mRNA production in order to do cell-free transcriptomic. And then I will finish with a comparison between the competition, in fact, for resources that there is between mRNA production and protein production. So first, description of the cell-free system. So cell-free system, when you do a cell-free reaction, it's composed of 3 elements, I would say, more than that, but we summarize them in 3 elements. The first one is extract. So I work with lysate-based cell-free systems. So we grow the cell, we extract the cytoplasm, and all the elements necessary for transcription or translation is in it. But then we supplement this lysate with a buffer, energy buffer, like amino acid, NTPs, to complete the composition of the cell extract in order to have an expression that run for a longer time and also a higher level. And eventually, because it's an open system, you can directly add your DNA. Here it's in the form of a plasmid, but you can also do it with linear DNA. So in the final mix, which is composed of the cell-free extract, the buffer, and the DNA, you have everything to run the metabolism, the transcription, the translation, and even the protein folding. So usually I work with superfolding GFP or folding fluorescence protein. So I don't really have competition at the protein folding level, but all the rest, there is resources that the different synthetic constructs that you will express in your cell-free reaction will compete for. And so when you do a cell-free reaction, so you prepare it in advance, and then when you are ready, you can add directly your DNA, and you will have your mRNA production that will be also degraded, because the degradation machinery is still present in the lysates, a part of it, at least, the one that is not connected to the membrane. And then you have the translation, and if your protein is not stable, you can have some degradation. But usually, also here with the GFP that I produce, you don't have the protein degradation. So how do we prepare it? We have those 5 different steps. The first one, so I usually work with E. coli, and we grow it to exponential phase in a rich media, but without glucose. Then we centrifuge to get the pellet. We wash several times with a buffer. We do a lysis. So I do it with a sonicator, but there is French press that you can use, or even chemical reactions to get rid of the membrane. Then you have a 1-hour runoff reaction that is supposed to let the ribosome finish the translation, so they are available in your mix when you start your cell-free reaction. But with some strains or some bacteria, it's not very useful to do the runoff reaction. Apparently, it doesn't help to improve the transcription or translation. And eventually, when you are finished with that, you centrifuge to get rid of the membrane debris, you take the supernatant and you supplement it with energy solution and buffer. And then you have all your transcription and translation that is working, and even part of the metabolism. So then what is really in the buffer? You have all the different elements that I put in bold here. And you don't really see, but most of them, in fact, all of them, they are dedicated to help the translation, the transcription, or the regeneration of ATP. Then two of them are quite more specific. One is a buffer used to maintain the pH around 7, and another one is PEG that is used to increase the density of the reaction, because the supernatant is quite liquid, and to increase the probability of the polymerase or ribosome to find their target, we increase the density. Then how does it look like experimentally speaking? So you can do your experiments in a append of tubes, but what we do is that we use 384 well plates. Some other people even use 1,536 well plates. And in the lab, we do reaction in 10 microliter, but depending on the plates, you can go from 1 to 20 microliter reactions. The advantage for us to do 10 microliter is more to avoid evaporation and also to fill the bottom of the well in our 384 well plates. The advantage with this kind of experiments is that you can do in parallel a lot of different experiments. So usually we do triplicate, or we even do 6 times the same experiments in one plate. And we always have a range of controls to compare plates from one to another. So how does it look like in terms of production of mRNA and protein over time? When you look at the transcription in a classical lysate-based cell-free system in batch, like I do, you have a 0 mRNA at the beginning, then you increase your production, you have a very short steady state of around 30 minutes to 1 hour, and then you have the degradation of your mRNA. For the translation, it's a direct link with this. At the beginning, you have a strong production, then a constant production during 30 minutes to 1 hour, and then your production decrease until it stops. And because in my experiments there is no degradation of the protein, but it stays the same until I stop my experiment. So I would say it's a time-constrained reaction in a batch culture, because after around 8 hours, depending exactly how you do your experiments, you stop it and you don't measure any more activity. The lysate that I use, it's E. coli-based, it's the most common one, but in fact there is more and more bacteria and even eukaryotic cells, so even with human cells you can do lysate, but me, I focus on microorganisms and on bacteria especially. And so there is already this collection of lysates that are possible. So the only limits to do cell-free is to have a growing bacteria that you can bring to exponential phase and have enough pellets to then extract sufficient amount of cytoplasm from it. And you need also to find a way to extract your cytoplasm without destroying the machinery, like especially the ribosomes, without damaging the ribosomes. So you have a translation that is actively working. But all those bacteria, there is paper describing protocols to obtain working cell-free reaction with it. So especially there is E. coli and Bacillus subtilis, so gram plus and gram minus bacteria. And now there is there is people working on more and more strains. So the advantages of cell-free is first that after you have done your extraction of your lysate, you have all your reaction ready. So you just have to add your DNA and your reaction will start. So it's a fast reaction to produce your protein. You don't need to transform because now it's an open system. It's directly the inside of your cell that is in this well, and so you can add whatever you want to it, as long as it doesn't damage too much translation and transcription. For example, if you put a chemical that is responding in ethanol, it will not work, but there is always a way to avoid this issue. It's not a GMO because there is no more living organism. You don't have interaction anymore when you express synthetic circuits with the living host, so no risk of mutation or burden because there is no more living cells. You can decouple effects. So in the previous work that I will not present today, I showed that you can separate, you can predict the different kind of burden, and you can use the cell-free in comparison with living cells to separate what I call metabolic burden for production burden, for example. And also you can add different parts of the circuits in different reactions to see how they affect your final products independently for each other. So it's quite useful to deconstruct the circuits or even something more complicated. It's also a low volume reaction. So like I told you, from 1 microliter to 20 microliters. So you can do a lot of experiments in parallel. You have higher level of control. So you decide which amount of DNA you put in it. You don't need different origin of replication because you just choose the concentration you work with. You can also add different type of plasmids because you don't care if there is the same resistance to an antibiotics or if there is potential competition between the origin of replication because there is no more divisions in the system. So you can co-express lots of different plasmids with the same backbones. And you can also control the composition of the reaction, and this is what I will show you later. Eventually, with some lysates, or if you supplement your lysate with some chemicals like games, you can work with linear DNA. So you don't even have to amplify the plasmid. You can directly amplify the PCR products and add it to your system, and it will work. A few examples of just applications. So the main one is the prototyping of synthetic circuits. But there is also work on self-assembly, like phages expression in cell-free, of production of therapeutics, like antibodies. And there is important works on recreating synthetic cells, of creating synthetic cells based on this lysate. So not starting from scratch, but reusing the cytoplasm of the cell, re-encapsulating it, and try to have systems that divide, reproduce, multiply in a tube from a lysate at the beginning. So a lot of works around cell-free. It's a very old technology, but with synthetic biology, there is more and more use of it. So now that you understand what is a cell-free system, let's talk about the work on maximizing protein production using active learning. So when I did this work and I wanted to produce more protein in a cell-free system without having it more expensive for the users, and especially, in fact, for each homemade lysate to be optimized, we explore the combinatorial space of different buffer composition. So here I put the list on the left of the different elements of the buffers that we modified in our final reactions. And for each of them, we used just 4 different concentrations. In red is the maximum one, so it was just the concentration in the initial buffer, and then we just decrease the concentration. And just with those 4 concentrations and the 11 different elements that we modified, we arrived to 4 million possible composition of cell-free. So we used here just one plasmid, always the same one, and we just move around and explore what's happening when you change the buffer composition. So the question was, is it possible to improve the protein production by exploring this combinatorial space? Can we predict what will happen after a little bit of training, what will happen if we change the composition and we try and we then measure the protein expression in this new composition, can we predict it? And can we find the parameters that are the more important and that are mainly involved in the protein production in cell-free? So first, we just to define the terms that I will use next, I will always talk about yield of protein production here. And what I call yield is just the fluorescence in a well minus the autofluorescence of the cell-free. In fact, naturally, it's produced some green measurements, fluorescence. And so I just remove that for the problems. And I divide it by the fluorescence of reference, which is the GFP expression I obtained in the initial protocol with the buffer composition of the initial protocol. So basically, it's all the elements of the buffer at their maximum concentration. So we were not, well, it's not possible for us to measure the whole 4 million different compositions. So we used an active learning approach to maximize the protein production. This work was done with by Mathilde Kolch and Jean-Loup Follon at INRAE. And what we did is that we started by just measuring 102 different compositions of cell-free to feed a neural network-based model in order to explore and optimize and maximize the protein to maximize the protein production. So the idea is to feed the model with data sets and then go in the right direction to maximize protein production. But we wanted also to have a good prediction to explore possible unknown space of our system to have a good predicator of what is happening, even if the protein production is not so good. So just to build the initial training set, what we did is that we used the reference buffer. Then we decided to go to some extreme. So we did first a collection of buffers with one element as the maximum, all the other at the minimum. Then the opposite, one element as the minimum and all the other as their maximum. And then we just set up a bunch of random composition to open up a little bit the initial training set. And so after, so what we did then is that we have this initial training set. We use it to feed the model. So we measure the output, which is GFP production, and we feed it to the model with an objective function of maximizing the protein production. So to do that, we use these neural network models that give us after new instruction to be tested, so new buffer composition to be tested. And we use an eco-acoustic liquid handler machine, which is a machine based on acoustic to distribute very quickly chemical compounds in 384 well plates. So we needed this machine because when you want to fill a full 384 well plates, you do around 4,000 pipetting volumes. And this is too long to do by hand because some reaction will have finished before another one even started. So we need a machine that can do it very quickly. And with the eco machine, we can do that in 30 minutes. Then put it at 30 degrees, wait for the fluorescence to be done, and then read it with a plate reader. So we did that for a few rounds. We increased our data set over time. And after a while, we obtained quite a high protein production. So our yield started at 1, and after 7 iterations, we managed to bring it to around 32, 33. And then we did a few other runs to check that we were at the maximum we could. So you can see that sometimes there is some yields that are very low in some iterations that are quite high in general. And this is because we force sometimes the model to go in some part of our combinatorial space that were really not well predicted in order to see if we were blocked in a local maximal or not. We will not be able to know if we are not a little bit stuck, but at least we managed to increase our yield 32 times. So this was more than enough for us. And after 7 iterations, we never managed to do better than that. We also use the 5-fold cross-validation to check that over each iteration we were better at predicting the next plates, the buffer, the different buffer, the link between the buffer composition and the protein production, and it was quite good after 7 iterations. We also use the mutual information score to find out which element of our buffer impacted the most the production of proteins. So here we can see that the magnesium, the potassium, the amino acid, spermidine, 3-PGA, and ATPs are the main elements that affect protein productions in our systems. And also, I don't show you here, but we tested lysate done by other people to see if it was always the same elements that affected the protein production, and it was the case. Another way to represent it is here. So you can see, so from green is low concentration, red is high concentration. Each line represents a compound of the buffer, and one column represents one experiment. So every column you have a composition, and this is connected to a yield. And here I just reorder the experiments in order to go from low yield to high yield. And what we can see with that is that, well, indeed, magnesium, potassium, and NTPs are really important for protein production. And what you can see here is that the higher they are, the better your production is. Then you have also the amino acids that are quite important for translation. And when they are quite high, it's better. And you have some elements like spermidine and 3-PGA that were fixed quite early in our iterations, but not at the maximum concentration. It seems that for them, it's an intermediate concentration that is the best. And then the other ones, basically, they are quite random. They don't really affect the yield in our systems. And in fact, on the experiments that we do now, we remove them and it didn't change the protein production anymore. In fact, this is kind of makes sense for some of them because, for example, the tRNA, we add the tRNA from E. coli in our mix, but in fact, it's already present in the lysate and it's recycled over time. So it makes sense that if you add more, it doesn't change anything. So to conclude on this part, we saw that we were able to improve the production of an homemade cell-free reactions without increasing the price. We provide a way to efficiently predict protein production, but after quite a large number of experiments. And we highlight the critical parameter involved in protein production, which are potassium, magnesium, amino acid, NTPs, spermidine, and 3-PGA. And in fact, this method can be used for improving whatever strain of bacteria that you want to work with. Now, the second part that we are doing right now, but for another reason. So the first part of maximizing protein production was to understand the impact of different compounds on self-reproduction, but also to maximize bioproductions. But here we want to maximize mRNA production to then move later to what we call cell-free transcriptomics, which is a way to use cell-free system to be able to measure mRNA level at the genome scales. And so be able to produce enough mRNA in cell-free to do RNA-seq and to explore the expression of genes in different genomes from different bacteria. But basically, the first step to do this kind of work is to increase the mRNA level. And for that, we use the same approach as the previous work, but this time we use a different plasmid. So we used ones that still have the GFP, but in addition, there is an aptamer, a malachite aptamer, which is able to transform an mRNA concentration in a fluorescence signal. So the GFP is green, and the mRNA, the malachite aptamer, which is called malachite green aptamer, in fact, fluoresces in red. And this time we use also an echo machine, but the new version of it, to do the same pipetting approach. So here we still have our buffer composition. We still have a lot of combinations that we can do. And so this time we explore a buffer composition still of around 1 million possible combinations. So we change the magnesium, the potassium, amino acids, spermidine, 3-PGA, NTP, phage, and the DNA amount. But in fact, the DNA amount very quickly arrive to the maximum concentration. We choose to stop the DNA amount at 10 nanomolar because for the next step of our experiment, we wanted to add genomes, and so we could not go to a higher concentration with the genome, so it was no need to increase the DNA too much. So this time, the work was done with Lea Wagner and Anne Orang at INRAE. And we used a little bit of a different approach to start our experiment and also to obtain the instruction. So to start, to define the initial set of conditions, this time we didn't choose ourselves where to start. But we use a Latin hypercus sampling, which is kind of the random one, but chosen we first distribute the area in which we will choose random concentration, so we are sure to be well distributed, but there is some kind of randomness in it. And so this was for us the best approach, but it's a choice that we made in order to be sure to not be blocked in a local maximum. Then instead of using a neural network approach, after some tests of Anne, we choose to go for a Bayesian optimization approach, which we will describe in the paper. And then we did the same system of active learning round with an instruction, distribution with the echo, measurement in the 384 well plates. And then we decide to put as an objective function to maximize the mRNA production, which is basically maximize the fluorescence of the malachi grain. This time we did some dynamic measurements. We have all the production of the times, and we choose to maximize the maximum peak. So in the model, in the Bayesian optimization, there is no dynamic data gave to it. It's just the maximum of the production of mRNA. So after 11 iterations, the same representation as before. Here you go from green to yellow to the minimum concentration to the maximum. Each column is a reaction and each line is a component. And you have here the magnesium, potassium, amino acids, pyrimidines, 3-PGA, NTPs, PEG, and DNA. And so the yield is calculated in the same way. You take the measurements in a well, you remove the autofluorescence, and you divide by the reference buffer minus the autofluorescence. And so over time, we managed to increase by 20 times the production, around 20 times the production of mRNA. And we saw that some elements are important to be increased and some of those are important to be decreased. So here is another representation of the same data set, but reorganized by increasing yield of mRNA. And so the first element is that the DNA is mainly at its maximum all the time. Some elements like PEG, spermidine are quite randomly distributed. But there is elements like magnesium and NTPs that are close to their maximum when you are in a high yield. The 3-PGA, again, is at the intermediate concentration, and the potassium, K-glutamate, is this time at the lowest concentration. So very quickly, the model decided to predicted that a decrease in potassium concentration will increase the mRNA concentration, the mRNA production. So in this work for the moment, what we can say is that we managed, like before, to improve the production of mRNA by 20 times in our homemade cell-free reaction. And we find, again, some parameters that are more important than others. And in these parameters, some of them must be very high, some very low, and other intermediate. And with a 20 time yield increase, we are now able to go to the next steps, that is this cell-free transcriptomics approach. So we didn't only measure the mRNA in this experiment, but also the protein production. And just to recapitulate, to compare the two projects, we have those similar elements in both. We have the the magnesium and the NTPs are both important for both processes. Well, the NTPs is important for protein production because you need more mRNA to produce more proteins. The magnesium we check in the literature impacts both the efficiency of RNA polymerase and ribosomes. But the amino acids, for example, is not really needed for mRNA production, and the spermidine seems to be just needed for the translation also. And for both systems, the 3-PGA is at an intermediate level, but the big difference is that the potassium, the K-glutamate element, that must to be very high for protein production and seems to be very low to maximize the mRNA production. And in fact, when you check the protein yield and the mRNA yield in all our iterations for this second work, because we have the GFP and the malachloride on the same plasmid, you can measure protein production and mRNA production in the same wells at the same time. And so you see that it seems that the more mRNA you produce, the less protein you produce. There is some example in which you keep some protein production even at high mRNA yield, but for most of the high yield of mRNA, you have low protein production. And the opposite, when you have high protein production, is with low mRNA production. And this seems to be the result of a low K-glutamate for our high yield of mRNA that seems to decrease the protein production in favor of mRNA production. So there is a paper recently published from Murray's lab in Caltech that point out that there is some energy competition between the transcription and the translation in cell-free, and this seems to be the case also for us. So now we have all those machine learning, active learning approach that give us a nice exploration of big combinatorial spaces, but we don't have so much understanding for the moment of why exactly an element affects mRNA production or the protein production. But obviously, there is some of them, like the amino acid or the NTPs, we know that they affect the system. But in fact, for example, the NTPs, they increase the mRNA production only if the magnesium increases. This is documented, but it's some effects that are not obvious at the beginning of an experiment. And so the next step that we want to move to is to complement this kind of approach with mechanistic model to have some explanatory effects and be able to perhaps find new properties which are not based on the objective function of maximizing mRNA or proteins. So our current analysis, in addition to cell-free transcriptomics, is to use the fact that active learning is a good way to explore a combinatorial space, to find very diverse combination with very diverse properties, with an objective function. So here our objective function was to maximize production of an element or another. And so it gives us a very diverse combinations and measurements with different really diverse compositions. So this is a good base to build mechanistic models that can be then tested in other areas that the model didn't explore because there is no need to go in those directions to maximize an elements. So the next step of what we want to do is that now we have these 650 buffer compositions that give us very, very diverse curve over times of mRNA and protein productions. We are building a mechanistic model to explain and to fit those curves, and then we want to do a last steps of tests with new compositions that will confirm or infirm the models that we build and potentially find interesting properties that nobody knows about in cell-free system. So in the future, it's an approach that we want to keep in the lab to have those an active learning for a given purpose, a real purpose, an objective like here, increasing mRNA to express full genomes, but also use these data sets to understand the cell-free system as his own research object of study. And so really understand, not just have a bunch of compositions that allow us to be very predictive, but we don't really understand the exact interactions that there are between them. And so use this diversity of buffers, this diversity of production to build a real mechanistic model. So this is something we are working on right now, and we'll see what we obtain with that. So to conclude, I would say that first, thanks for listening to my presentations. I want to thank the cyber team at Michaelis that I work in, which is led by Mathieu Jules. I also work with the BRS team led by Jean-Loup Follon, still at Michaelis. And we also have the help of Miguel and Mayaj, which is two other units, but on the same campus in Jouy-en-Josas. And with all of these different people, we managed to have good data results for active learning and self-test systems. And also another thing is that right now we are building what we call a self-free biofoundry at INRAE in order to automatize the measurements and the production of self-free systems. So if you are interested, you can contact us. If you have some idea of something to do or some construct to test in cell-free, we are open to discussion, because we are right now building this biofoundry to test new systems and challenge our approach. So thank you very much, and I'm listening to questions.
Okay. Thank you so much for the talk. I think, yeah, we have a couple of questions in the chat box, and I think I'm going to start from the first one. So from Olivier, how smooth is the high-dimensional landscape of compositions? Do you expect that there are multiple optima, and can the Bayesian optimization approach pick up on this? I think that comes from the part of optimizing mRNA production.
Yes, yes. But that, in fact, I have no. I don't really. In fact, the only way to know from me, from my perspective, so I'm not a mathematician, so it's more a biological approach. But I will say that the only way I will know that I'm not stuck somewhere and there is perhaps better optima somewhere else is by building this mechanistic model to really understand what is happening with the modification of those different compounds. So I would say that for the moment, I don't know if there's no other optima somewhere in my buffer compositions. If I cannot reach the same maximum mRNA levels, or even a little better ones somewhere else in this system, because I tested 650 buffers in 1 million ones, so potentially there is perhaps somewhere one that is a little better.
Yes. Okay. Another question is also from Olivier is, how much of an effect does the volume of the cell-free container have on the protein and mRNA production processes? So how would you scale the production?
So I know that there is people trying to scale up cell-free reaction to go to liters, milliliters to liters. I know that one of the issue can be I don't know if it's exactly the question, but I will answer that. And if you want more details, tell me. But the oxygenation and the distribution of the compounds in the cell-free reaction can be quite complicated to maintain homogeneous in a very, very big container. For us, it's not an issue at all because we are in 10 microliter. we agitate a little bit our reaction. So we never face an issue with that. But I suppose that when you go to very high volumes, especially the oxygen availability and that sort of problem will appear. And that I don't know how to manage.
Okay, yeah, thank you. So another question from Hannah. Have you tested protein production of any other proteins? How general do you think are the optimized conditions for production of GFP?
So this is another work we have in mind. For the moment, I just tested GFP. So I think what I think is general can be generalized is the approach, the active learning approach. But I don't think the composition will be the same for any proteins. So I would say that what I advise is that if you want to optimize another protein, use the same approach, but not the same buffer. But I think it can be, what can be interesting is to use those buffers as a starting point, not restart from scratch. So we will put all the data on in a file. We'll write a data paper with all the data as raw as possible, and so everyone can use that as a starting point.
Okay. Thank you. So we have more questions coming up. So from Ian, first one, how does cell-free protein production rate compare with cellular production rate? And the second one is, in an earlier slide, you showed that mRNA concentration decreased due to degradation. How would it be possible to remove ribonuclease from the cell-free system to eliminate degradation?
So when we compare with, for me, it's quite difficult to compare the cell-free production rate with the cellular one. In a way that it's very, very concentrated what we have in the lysates that we extract. In fact, it's liters of cell culture that are concentrated in milliliter of lysate at the end. So I have an issue with my, I don't know if you can see it. So I would not be able to quantitatively compare the two systems. It's something that must be done. I agree. And for the mRNA decrease due to degradation, we try to add some elements to inhibit the ribonuclease, but it didn't work. In fact, it blocked my. When I added the inhibitor, it just stopped the production. So I don't know. And then I also used the strain with to make my lysate a strain without the ribonuclease, but in fact it was ribonuclease linked to the membrane, I think, and so it didn't change anything either. So there is some work to do on that. Just to tell you, there is some buffers that I tested that seems to decrease the degradation over time. So in fact, perhaps already in the buffers that I tested, there is some of them that seems to do this job, and I have to go deeper in the analysis to find out which one.
Okay, yes, thank you, Olivier. And question from Tom. Really interesting talk. Thank you. Yeah, I think we all agree. And have you compared the protein yield to those from commercially available CFE kits, such as Pure Express?
I didn't compare with Pure Express, but for the first paper, we compare with there is this company involved with Vincent Noireau's work, which use lysates, Arbor Bioscience, I think. And in fact, when we compare our reaction with theirs, at the end of our optimization, we were at the same levels. So in fact, we managed to bring our homemade system to the same as a commercial kit, but not so much better. So it was, I think that they probably optimized also a lot their kits on their side. And so we managed the same. And for the pure express, I never used it. I know that it's a purified protein instead of a lysate, but it's something that could be interesting. And just also, just to finish, for the protein production, we did the comparison, but for the mRNA production, we didn't manage to do it because in the buffer, the kits, the commercial kits, there is DTT in the buffer, and this creates issues with the malachite. In fact, when you mix malachite and DTT, it doesn't work anymore. So you cannot measure a fluorescence level. So if we want to compare the kit, we have to do some qPCR. So for the moment, we haven't done it, but we'll probably do that at some point.
Question from Hannah. Have you tested different temperature increments?
Sorry, I had I moved my computer. If I tested different temperature, no. I always do all the experiment at 30 degrees. So I didn't try testing other temperature.
Yeah, I think final question for now from Olivier. What kind of mechanistic modeling approach are you exploring? Do you think something like the oxygen absorption could be sensitive to composition and determinant of the yield?
So for the moment, we are really building the mechanistic model. So we started with something really simple with a Michaelis Menten-like explanation of the relationship between mRNA production and NTP, and the same for protein and amino acids. And we try to find out which parameters influence the Vmax and the Km. So we really start with something simple, and we slowly increase the complexity with something simple like production of mRNA, production of protein, degradation of mRNA, and that's it. For the oxygen absorption, but for the moment, the only test I have done to check the impact is just by comparing a plate which is sealed with a plate that is not sealed, a plate that is shaking, and a plate that is not shaking. And with my 10 microliter reaction, I haven't seen any change in the yield. So for the moment, I don't consider the oxygen absorption as an issue that could be added at some point.
Okay, yes, thanks. And also we have Tom saying thank you. Yeah. And any more questions? You can raise your hand.
I do have a question. It's more about the model. So are you eventually thinking of adding some of these, what you learn mechanistically, like the mechanistic models, back into the machine learning models as a priors maybe or something? Like, is that something? So you're going to have the machine learning models that do active learning, and then you have that will give you insight on the mechanistic models. You could eventually also think about adding some of these mechanistic into the machine learning.
Yes, but for the moment, it's not the aim of what we are doing, but it could be interesting to relink the two after. We will see. We will see, depending of how good we are at explaining things. If we go back to that, but it's interesting. It's something I would keep in mind. Yes.
Because you could have hybrid models. It's not purely machine or not purely mechanistic.
True. So for the moment, it's true. It's another approach that is very interesting, but I think it's something I could think about even before starting the round of active learning, because, for example, the NTP, I knew how it interacts with the polymerase to produce the mRNA. And even if I didn't know about the other compounds, I could have started with an hybrid model at the beginning. But now it's true that we explore so much that I don't know if we will go back to, but it could be interesting. In fact, instead of exploring new buffer based only on the mechanistic model, we can explore new buffer based on the hybrid model. It's true. Yeah, it's interesting. Yeah, I I will talk to Anne about that. Thank you.
Also, another question, sorry, about the model. I don't know if anyone has asked a question, but so right now it's training basically on having, I mean, it's training on one output. Would you see, I don't know, but will someone eventually be interested of maximizing maybe two different outputs, and is that possible?
Yeah, sure. But it's something, but in fact, for more complex circuits, not just maximizing the production of final elements. For example, we are working with Jean-Luc Follon with the work on perceptron. And in the perceptron, we want to optimize profile of different combination of circuits, not just one output. And Anne Wang is actually doing that. So she's using the same active learning approach to with an objective function that is way more complex. That's what I'm doing right now. And it seems to work. The only point is that when you look complex profiles, you need more experiments. And so you need a little bit more runs to learn. So for me, it was easier because I can quickly do 100 by 100 experiments. And so feeding the model was easy. But the moment you complexify your output function, you start also to increase your number of experiments. But it's totally doable. I mean, there is no problem. It's just it's what we see with this perceptron approach that we try to optimize. You need way more than 600 experiments to optimize it.
Okay, so no more questions from the audience. Yeah, so thank you so much.
Thank you.
Yeah, it's really nice to have you here. You can see like the audience have really a lot of questions, and yeah, it's really interesting topic. And yeah, thanks again for sharing this with us.
Thank you very much for the invitation, and now I have new ideas to test. So it's good. Good. Thank you very much.
Thank you, everyone.
Thank you.
Have a good evening.
You too.